So in this final video for this next lecture about G protein coupled receptors, we are going to talk more about different G proteins that bind to G protein coupled receptors. So you're going to learn about 4 more important different types of G proteins, and we'll introduce the downstream targets. So this is kind of where we stopped before. We have G protein coupled receptors that bind different G proteins. We very briefly talked about its trimeric, G alpha, beta, and gamma. This binding is going to activate them by exchanging GDP that's bound to G alpha to GDP. That binding of GDP will lead to their dissociation, and then alpha subunit is going to activate certain effectors and beta gamma different effectors, and then they are going to be deactivated and return to the normal state. So we are going to be talking more about what different G proteins we have, what different trimers, and what are the downstream signal that goes from both of these proteins. So G protein coupled receptors are going to activate one more G proteins, and the function of G proteins is to transmit information about the concentration of neurotransmitter that's outside of the cells to the change in different activity of different enzymes and then secondary signaling molecules. So that's what they do, they're transmitting this information. So what's important to know, that G proteins are trimers. They have pretty much, they're trimers, but they have 2 functional units. One is alpha subunit, and then there's beta gamma, which are very tightly bound. So when they're inactive, they're binding GDP. So this is their inactive state. We have all subunits, so alpha, beta, gamma are bound together. When GDP is exchanged for GDP, this activates. So when alpha subunits binds GDP, this will dissociate them. So this is active state, which is then dissociated into alpha subunits with GDP and beta gamma subunits separately. And then each of these can lead to downstream effect. Then this GDP is going to be hydrolyzed. It has alpha subunits in itself, actually has GDP as intrinsic activity, but as we will see later on, it can also be helped. When this GDP is hydrolyzed in GDP, it's going to increase the affinity back for alpha subunits to bind beta gamma. It's going to go back to the inactive state. So this is the cycles through this G proteins go. So again, it's trimeric protein, that's what you need to remember. They are together when alpha, beta, and gamma bind. When alpha binds GDP, it increases affinity for beta gamma. Exchanging GDP activates them, and then they split, and each alpha subunit by itself and beta gamma can exert different effect. That's when they're active. We are going to talk here about several different G proteins. So we can, and this is mostly based on their alpha subunits. So first that we are going to talk are alpha subunit S and alpha olfactory, because S stands for stimulatory, that we've told before, because it stimulates activity of adenyl cyclase. Activated adenyl cyclase is going to lead to the production of AMP, so that's its downstream effect. It's important to foreknow to them that they're sensitive to chlorotoxin, and that's how they have been separated by the other ones. Second group are alpha I, inhibitory alpha, or alpha O for other. They inhibit adenyl cyclase, so that's their activity, and therefore they decrease the amount of CMP produced in cells. These are sensitive to pertussis toxin, so that's how we can separate them from the other alpha subunits. In addition, beta gamma subunits from these, because they're really abundant, are going to have their own effect. They're going to either cause open or close channels, but as we'll soon see, they can do some other effects as well. Then we have alpha Q, alpha 1114, and gostosicin, which is the one that plays role. You will see in the taste we have many different alpha, but their basic activity is to activate phospholipases, which will then lead to production of inositol-3-phosphate and deacylglycerol, and also increase calcium, cytoplasmic calcium levels. And finally, the fourth is the transducin, alpha-T, which is very important in the vision. In this case, it's going to activate cGMP phosphodiesterase and decrease amount of cGMP in the cells. So these are, again, major ways through which G proteins work. We have alpha S stimulates or activates adenyl cyclase, alpha I, which inhibits adenyl cyclase. We have alpha Q, which leads to activation of phospholipases and increase in IP3, calcium, and deacylglycerol. and transducin, which activates cGMP phosphodiesterase. And again, we will talk now about each one of these pathways in more detail. So first one, again, alpha S or alpha factory as well. You have to remember just that alpha S, because S stands for stimulatory, activates adenyl cyclase. So this is your alpha S G protein that has GS. You can call it GS, or alpha S is the subunit, that's why it's given whole name GS. So when transmitter, in this case we can call it norepinephrine binding beta adrenergic receptors. When norepinephrine binds it, then they are going to lead to conformational change, which is then going to increase their affinity for GS or alpha S in complex with beta gamma, which is then lead to the exchange of GDP for GDP. Then alpha subunit separates, alpha stimulatory separates from beta gamma, goes and binds adenyl cyclase, which is then going to lead to the increase in level of CMP. It's going to then dissociate and activates. Once the GTP is straddlized, the GDP is going to dissociate, and then it can bind again if this neurotransmitter is still bound. The bottom line is we are going to lead to activation of adenyl cyclase and increase in CMP levels, which will then lead to increase in protein kinase A activity and increased phosphorylation. These are your beta adrenergic receptors, your dopamine receptor of type 1, which are dopamine receptor 1 and 5, purinergic receptor of A type, A2A, and serotonin receptor 5-HT1. You do not need to memorize which one of these are using what. This is just for your knowledge if you want to know that. Let's say they activate adenyl cyclase. Adenyl cyclase is transmembrane protein, so it's already present in membrane, and it is going to again associate with alpha stimulatory, catalyzing reaction from ATP into cyclic AMP, and it's called cyclic because it has this cyclic ring feature. What is really important to know, while adenyl cyclase makes CMP, there are also enzymes called phosphodiesterases which cut that bond and make it into AMP. So that's the way that the signaling can also be decreased by increasing the activity of phosphodiesterases. You do not need to memorize this, but I want you to be aware that there are different adenyl cyclases that can be expressed in the cells, and these different adenyl cyclases are differently regulated, which will tell you that depending on which isoform you are expressing in your cell, you may have different level of increase in CMP activity. For example, adenyl cyclase of type 1, which are 1, 3, and 8, need these two, they're called coincidence detectors, so they actually need stimulation not only by GS or alpha S subunit, they also need calcium increase, calcium bound to calmodulin, so need both of these things to happen for their full activity. On the other hand, if you have adenyl cyclase 2, 4, and 7, they will need this alpha stimulatory subunit to bind, but also beta gamma subunit will lead to their activation. Adenyl cyclase 5 and 6, they just will be activated by alpha subunit, stimulatory alpha, same with adenyl cyclase 9. However, if you are either 5 or 6, if concurrently you have activation of GI or O, so inhibitory, they can actually inhibit this activity. So the level of their activity will be determined by how much of each one of those is activated. So when you have things like that, when activity of the enzyme is regulated by two different pathways, these are called coincidence detector because they will integrate stimulation of different neurotransmitters or different receptors to which they are binding to produce the final effect. And then we talked that downstream effect of, again, increased levels of CMP, which can then lead to the activation of protein kinase A, which we have the whole class about kinases in which we will talk much more about protein kinase A, and you don't need to memorize all different downstream targets of protein kinase A, but you should memorize that these changes in protein kinase A activity or phosphorylation by protein K can lead to this modulate your cell function over broad temporal range, then volume, because again PK will then exert much further away from your receptor, and it can coordinate many diverse cell function because it can affect synaptic vesicle proteins, ion channels, enzymes, etc. In addition to, again, activate pKa, CMP that's produced by active adenyl cyclase can go and bind the channels. In this case, this is in alpha-factor H transduction. The CMP is going to bind the channels and it's going to open these channels through which calcium and sodium can enter, which will then lead to depolarization. And again, as I told you, these have been identified initially because they are inhibited by choleratoxin. They're actually not, choleratoxin prevents GDP to be removed or hydrolyzed into GDP, so it leads to persistent activation of adenyl cyclase. So it prevents its deactivation. So you have consistently active this pathway, which will then on its own cause many problems. The second pathway that we want to talk is alpha I's, so GIs, or in which alpha I subunit is active, or alpha O stands for other again. Both of these are going to lead to inhibition of adenyl cyclases, decrease in CMP levels. Again, it's prone here for dopamine 2 receptor, which is bound to GI, decrease activity of adenyl cyclase, decrease CMP, and decrease phosphorylation of protein kinase A. This is your D2 receptor group, D2, 3, and 4 receptors, alpha adrenergic receptors, so beta adrenergics are stimulatory, but alpha are going to be inhibitory, the same way as D1 receptors are stimulatory, but D2 are inhibitory. Muscarinic acetylcholine receptor 2 and 4, somatorenergic receptor, quite a big number of serotonin receptor, GABA B, mGluRs 2 to 8, so most of mGluRs are going to have 2 and 3 are GO and the other one are GI, and cannabinoid receptors CB1 and CB2. Now, because alpha subunit has really low affinity for adenyl cyclase, GI proteins are really abundant in the cells, and therefore they are going to lead to production a lot of beta gamma. So most of the effect when we talk about beta gamma will be through the GIs or proteins because they have sufficient levels of beta gamma to go and then exert its effect. Now, beta gamma, as we talked, can lead to the opening of Kirk channels and hyperpolarization. It will also lead to the inhibition of calcium channels, both N-type and PQ type, but to different levels. It can also affect the activity of phospholipases, and as we have seen before, beta gamma can either activate adenyl cyclase from one type, or they can inhibit the adenyl cyclase of the other type. Pertussis toxin is the one that affects GIs or G-alpha I because it actually prevents activation. It's the same mechanism, so it's again binding of ADP ribose as the chlorotoxin does, but this time it's to the ADP bound or non-active form of GI, and therefore it prevents its activation, and therefore adenyl cyclase will remain active more. Then we have GQ. Again, we told there are other G proteins, G11, 14, that does function through the same way. They activate phospholipases. In this case, we have phospholipase B. That will catalyze hydrolysis of phosphatidylinositol for 5-bisphosphate present here in the membrane, so it's phospholipid in the membrane. It will lead to the production of diacylglycerol, that again is staying in the membrane, and also production of inositol 3-phosphate. Inositol 3-phosphate can go to the plasmatic reticulum, and then it's going to bind inositol 3-phosphate receptors on the endoplasmatic reticulum and lead to the opening of the calcium channels and leakage of calcium from the endoplasmic reticulum into the cytosol, and that way we have significantly increased calcium concentration. Calcium and diacylglycerol together can lead to the activation of protein kinase C, or calcium by itself can lead to the activation of calcium calmodulin kinases, CAM kinase 2, 4, depending on which place in the cell we are. These are going to be your serotonin receptor. These are some examples. Your group 1 mGluRs, 1 and 5. The other one we saw we are leading to inhibition or activation of GIs. And we have a few of acetylcholine receptors. It's important also to know the phospholipases can be activated also by beta gamma subunits from the GIs that we have told, because these are the ones that will have abundant levels of BQ released. In addition to production of deacylglycerol and IP3, decrease when we activate GQ protein receptors, GQ proteins, this will also decrease levels of PIP2. And this decrease in PIP2 will actually lead to the closure of certain M-type potassium channels. So these M-type channels, in order to be open, need to be bound by PIP2, phosphonosidyl biphosphate. When we have activation of, let's say, acetylcholine receptors that bind these type and activate GQ, it's going to decrease the amount of PIP2, and that's going to close M-type channels. Closing of these potassium M-type channels is going to decrease the efflux of potassium that normally happens, leading to this depolarization, which is otherwise known as slow EPSP. As I have mentioned before, some of your taste receptors are going to be GQ, so transducin, some of the transducin, that's the name of the G proteins that are associated in this case with the bitter taste, are going to lead to activation of phospholipase C, IP3 production, and increase in the calcium levels. Also the one that will taste sweeteners works in similar ways. The other taste receptors, such as one for sugars, are actually working through GS, which will lead to increase in CMP levels. But quite a few of them are going to lead to increase in phospholipase C and calcium, and also we have some that lead to activation of phosphodiesterases, which is similar to what we see opposite, or transducin cycle. So this is again in vision. So we have again, this is a different function, transducin, which is a G protein coupled with the opsins, are going to lead to the activation of phosphodiesterase. So that's their primary factor. Phosphodiesterases are the enzymes that are going to decrease levels of cGMP to GMP. So how this works in vision? So normally when they're in dark, there are lots of production of cGMP. The cGMP is going to bind to the sodium channels and keep them open, and then that way lead to depolarization. When there is light, light is going to lead to the change protein absorption or light absorption will lead from retinal from 1-cis to trans-retinal, which is activated. So this activates G protein coupled receptors, which is rhodopsin in this case. This activation, as we talked, lead to activation of G proteins, in which calls, in the case of vision, is called transducin. Transducin activation again leads to exchange of GDP for GDP. Activated G protein or transducin will lead to the activation of cGMP phosphodiesterase, which is going to cut cGMP and then decrease levels of cGMP, make it into GMP, and lead to the closure of these channels and then hyperpolarization. How is this the signaling and then at the level of G proteins? Remember I told you this when we had the first time introduced activation, we had those time clocks on G proteins. Once alpha subunit binds GDP, it has its own GDPase activity, which is going to cut this GDP and have GDP bounded. In addition to its own GDPase activity, this can be stimulated by some of the effectors, and some of those effectors, such as adenyl cyclase, are going to stimulate GDPase activity and then lead to the hydrolysis of GDP into GDP and phosphor. In addition to some effectors activating this, there are proteins called RGS. This stands as regulator of G protein coupled signaling proteins, which can enhance this GTPase activity and then lead to the fastened decrease from GDP into GDP. And that way turn the G proteins into the inactive state. So this is the protein that I want you to think of at home. Problem: stimulation of the vagus nerve releases acetylcholine, which binds muscarinic receptors 2 and 4. That causes hyperpolarization and slowing of the heart rate. This is again the example of this. So we have hyperpolarization and slowing of the heart rate. Can you provide what you learned about muscarinic receptors 2 and 4, how they work, how could this lead to hyperpolarization?